Good evening, everyone. Good evening. Let's wait for the students to join in, then we'll start off with our lecture series. Okay, so today is a new lecture series that we are starting on Botany Insider, and from today onwards, in this complete week, we'll be having five or in this week we'll be having 5 concept mapping free lecture series. Okay, so make sure that you join in every single day at 8 p.m. on Botany Insider. Okay, so let's wait for students to join in and then we'll begin with our new lecture series on concept mapping, right? Good evening to all the students joining in the class. Good evening, Mayank. Good evening, Niharika. Let everyone join in, then we'll begin with our class. Okay, let's begin. So before starting the video, if you're new to our channel, make sure to hit the subscribe button. We are very close to 10,000 subscribers. Let's hit this number very soon. In addition, download the application from the Android as well as from the iOS. We have a lot of courses running for you all. In addition, this is the new course that is running for the time being, which is related to the structured revision. So this is a crash course 2.0 for the students who are finding a difficulty in structured and organized revision. You can join us in this particular course. You'll be getting PYQ pointers hard copy delivered at your doorstep. In addition, every day at 8:30 early morning we have our classes. So make sure if you wish you can enroll in here. In addition, we also have in addition, we also have a new course running which is for the June 2023 itself, but it's a full course, a full detailed course where you'll be getting a hard copy notes. So, depending upon whatever is your requirement, if you are in revision phase, go for the crash course. If you are in the detailed phase, you wish to have detailed explanation, you can have the full course. Okay, in addition, we also have the mock test available. So 40 plus CBT mode in the similar manner, the way CSIR takes up the test in the similar interface, we have these tests available for you all for practicing out and for revising the concepts very easily. Okay, so and in addition, if you wish to have the notes booklet, you can download the app and place your orders for the notes booklet as well. This was about all the contents available for you all. Now let's begin with our class. So if you all have seen this particular, the name of this particular series is concept mapping. So what is the main agenda that we are going to take up in this particular complete marathon is depending upon whatever the response of students is, whether they are liking this particular thing and it is fruitful for them, we'll move in the similar manner. Okay, so good evening, Selva. So now, talking about concept mapping, what we'll be doing in this particular lecture series is, for example, today the topic for today's class is central dogma of biology, right? So we'll be understanding about every single thing related to central dogma. We'll have a quick discussion regarding what is it all about, what are its various components, what are its important features that we have to keep in mind looking into the time shortage. Like depending upon whatever time we have, we'll go in the similar depth. So if you wish to have a complete revision for the same, we have the crash course. If you wish to revise from here itself, you can go from here itself as you wish. So here we'll be pointing out only like surface pointers depending upon the time. right? So let's begin with our class now. And then we'll be solving out one or two previous year questions as well. So make sure everyone is staying in the class till the end, because at the end questions would be solved and it would help you to implement your knowledge into the questions asked as well. So it would be a full, like, complete set of a specific topic getting revised over here. Now let's talk about increase the volume. Please check from your end. From my end there is 100% knowledge. The volume is 100% over here. Please check from your end once. Just try to increase the volume of your mobile phone or laptop, whatever you are using. Let's talk about the central dogma. So what is central dogma of biology all about, right? So when you look into unit number 3, that is fundamental processes, over here in that particular lecture series or in that particular unit, we look into various mechanisms getting solved. For example, okay, it's okay now, that's good. Okay, so talking about the central dogma of biology, it says that you have the DNA which gets converted into the RNA, and then the RNA gets converted into the proteins, right? This is what the central dogma is all about. So the DNA is carrying the actual information in the forms of the genes, but the cell cannot take the dictation from the genes directly in order to get any specific work completed, right? So what is required? These genes need to get the information in such a manner that it could be easily read by the cells. And for cells, it is the proteins which are of the easiest language, and the cell can easily take up the command from a protein. So for that matter itself, the DNA gets converted into the RNA and RNA gets converted into the proteins. Let's look into each of these things now. First thing first, the DNA copy number need to be increased in order to make sure that from the first generation to the next generation, the DNA is getting copied. So for that, what is required? There need to be increase in the number of the DNA copies. So for that, what is required? For that, you need to have DNA replication, right? So DNA replication is what? It is a process via which the number of the DNA molecules get increased in the cell. When we look into the DNA replication steps, so there are 3 basic steps. Step 1 is initiation, step 2 is elongation, and then we have termination, right? So these are the 3 major steps. Yeah, so these are the 3 major steps. At each of the step, there are various proteins, there are various enzymes working over here, and in DNA replication, most important enzyme is DNA polymerase. Depending upon whether it is prokaryote, depending upon whether it is eukaryote, the type of the DNA polymerase differs. For example, in the prokaryotes, you have DNA polymerase 1, 2, 3, 4 and 5. In case of eukaryotes, you have alpha, beta, delta, all these ones, right? So remember about the different type of polymerases. They have difference in their functionalities as well, and they have difference in their structures as well. So make sure that you are revising all these things. Second step over here is that after the DNA replication gets done, and because it is a mechanism, there are chances that it could lead to some kind of problems as well. There could be some kind of backlagging as well, in terms that it might get लाइक वो गलत तरीके से कॉपी हो जाए, फर्स्ट थिंग. सेकेंड थिंग, वहाँ पर कोई मिसिंग अवेलेबिलिटी हो किसी पर्टिकुलर न्यूक्लियोटाइड की, सेकेंड थिंग. थर्ड थिंग, देर आर चांसेस कि कोई पर्टिकुलर पार्ट चेंज ही ना हुआ हो. सो फॉर दैट वी हैव अ प्रोसेस कॉल्ड एज DNA रिपेयर मैकेनिज्म्स. सो डिपेंडिंग अपॉन कि क्या प्रॉब्लम हुई है DNA रेप्लिकेशन के अंदर, उसके बेसिस पर हमारे पास बहुत सारे मैकेनिज्म्स होते हैं. देर आर अ लॉट ऑफ मैकेनिज्म्स वाया विच द DNA रिपेयर गेट्स डन. So it could either be base pairing repairing, it could either be nucleotide repairing, etc, etc, right? As in when the DNA gets replicated and then repaired properly, DNA को अब हमें RNA के अंदर convert करना होगा. For converting the DNA into the RNA, there is a process which is given the name of transcription. So conversion or the copying of the DNA into the RNA format, it is called as transcription. Over here, remember one thing, the DNA gets converted into RNA. The DNA which gets converted into RNA, it is not in final process form. It is in the pre or it is in the primary stage. So when it is in the pre-mRNA stage, it needs to be processed into the final mRNA. For that you need to have RNA processings being done. Now हमारे पास तीन टाइप के RNAs होते हैं. We have rRNA, we have mRNA, we have tRNA. Transcription के अंदर there is a important enzyme given the name of RNA polymerase. Depending upon हमें कौन सा DNA, कौन सा RNA फॉर्म आउट करना है, हमारे पास डिफरेंट RNA पॉलीमरेज का रिक्वायरमेंट होगा. फॉर एग्जांपल rRNA is via RNA polymerase 1, mRNA is via RNA polymerase 2, and tRNA is via RNA polymerase 3. Remember about this, it is very, very important. Then after the processing, now in this processing, what is required? There are various steps, there are various important types of processings depending upon which RNA it is. First one is capping, right? Then we have the next step as tailing, right? And then you have one more major thing which is known as splicing, because हमारे पास जो transcription होगा, whatever organism that we are looking into, the organism could either be prokaryotes or eukaryotes. In the prokaryotes we do not have introns being present, लेकिन eukaryotes के अंदर introns का presence होता है, and we need to make sure the introns gets removed off and the exons are clubbed together, जो splicing process के अंदर होगा, right? Now after it gets converted into the final processed mRNA, now what happens, RNA को हमें protein में convert out करना है. Now this particular step is called as translation. That means final जो step होगा, it is translation. Over here as well you have three steps: initiation, elongation and termination. तीन steps वापस repeat होंगे, जहाँ पर there are a lot of initiation factors, there are a lot of elongation factors and release factors depending upon whether the organism is eukaryote or prokaryote. उसके बेसिस पर we have differences in the type of the factors being used, and after the translation or in the translation process again you need to have a lot of components functioning over here. And finally last में क्या होगा, because हमारे पास अब जो प्रोटीन है, दिस प्रोटीन इज मेड अप ऑफ व्हाट? प्रोटीन इज मेड अप ऑफ अमाइनो एसिड्स, राइट? एंड अमाइनो एसिड्स are the एक्चुअल बेस लाइन जो हमारा सेल आइडेंटिफाई कर सकता है, विच आर सेल कुड एक्चुअली आइडेंटिफाई एंड अंडरस्टैंड अबाउट इट. सो that is the reason why the proteins are the final results, and these proteins are taken up or they are used as a dictation by the cell. Our cell उसी के according cellular response देगा, whatever is the requirement. In the similar manner translation complete out होगा. Just check whether everything is clear to you or not. I have tried to add on like every single point over here. Just take a minute, get through all the pointers that I have explained. If you have any doubt, you can ask quickly. Everyone, just check whether everything is clear or not. Clear everyone till this particular point? Yes. Selva, Mayank, Niharika, Sarbeshwar. Just check, bache, is everything clear or not? If yes, then we'll proceed further with the questions and some more important discussions. Others also? Okay, so now talking about the important pointers. So with this particular topic you can understand that DNA replication first, then you have DNA repair as the second thing, then we have transcription, then we have RNA processing, then we have translation, and there are various other pointers regarding capping, tailing, splicing, all the mechanisms of various steps that I have already told you. All these things are very, very important. I have given you a complete concept map of the central dogma of biology. Now if you wish to revise it from your end in this particular manner itself, you can go ahead, and if you wish to, like, you're not able to revise it properly, you can join us in the crash course. Okay, so this was about the complete central dogma. Now the complete central dogmas concept mapping. Tomorrow also we'll be having a very good discussion regarding a different topic. You'll get the notification for the same early morning tomorrow itself. So let's solve out few of the questions now, so that you can try out whatever you have revised in your revision time, whether it is clear to you or not. Okay, so this is the very first question for you all regarding the 5' cap stability. So question is that what is the requirement of the 5' cap of the RNA? So you can correlate that this particular point is in relation to the RNA processing, and specifically if we look into it is mRNA processing. So try to find the answer for this particular question, everyone. Yes, try to find out the answer for this question. It's very simple. If you have revised it properly, you'll easily get the answer. Try to find the answer for this. Yes. Okay. Others also give it a try. Sayed's Sadav is also there. Who else? Selva is there. Sarbeshwar is there. Okay. Let's check the answer for this. The answer for this question is option number B. Very good, Niharika. Niharika has marked the correct answer. Answer is stability and the transport of the mRNA. So 5 dash capping in the RNA is required for both the stability and the transportation of the RNA. Remember about it, it is not just stability and it is not just transportation, it is both stability and transportation. Remember about this. Let's move forward. This is the next question for you all. Try out the question. Question is, origin of replication usually contains which of the following repeats? First one is GC rich sequences. Second option is AT and GC rich sequences. Third is no particular stretch. Fourth is AT rich sequences. Try to find the answer for this question, everyone. Give it a try quickly. Niharika, read the question carefully, bache. Question is asking, origin of replication usually contain which of the sequences? Read it again, everyone. I am repeating the same thing. Read the question again. And also, there is a very important logic behind it, which you need to remember. Let's check. Answer is option number D, that is, it need to be AT rich. There is a very important information regarding this, and that is, between A and T there are 2 bonds, between G and C there are 3 bonds. So between A and T, because 2 bonds are there, so there is requirement of lesser energy as compared to GC rich region. That is the reason why we have A and T rich sequence being there, which is usually present in the origin of replication area. Okay, so make sure you remember about this. And Niharika, just check the question. Shadow boy Mayank, you could have read the question again. And Niharika, you are there in the crash course as well. Read, that's why I am repeating this. Try to read the question again, bache. Question is, origin of replication usually contain AT rich region, not GC rich region. Okay, next question. Now this is the next question for you all. Question is related to type 2 splicing. So when we look into the splicing, so there are 2 groups of intron, group 1 and group 2, right? So you need to find out that in the group 2 or the type 2 splicing, which of the following is correct. So there's a very important mnemonics that we have discussed. So try to find the answer for this. First option is a GOH from the outside makes a nucleophilic attack on the 5-P of first base of intron. Second is a free 2-O of the internal adenosine makes a nucleophilic attack on the 5-P. Then we have 3-O of the internal adenosine makes a nucleophilic attack of 5-P. And last is the hydrolysis of last base of exon is carried out by U2, U4, U6. Get the answer for this question, everyone, quickly. Give it a try. Everyone, quickly mark the answers. Done, everyone? Quickly, everyone, read the question properly, get the answer. You won't be getting a second option, like second chance in writing down the answers in the examination. So make sure that you are reading the question carefully. Sorry does not work in CSIR examinations. Misreading is something which you need to take care of. Okay, and it is for everyone, not just one student. Let's see, answer for this particular question is option number B. So in the type 2 splicing, what do we have? If you remember, in type 1 there is external attack of 3 OH, and in the group 2 the internal intron, the intron is there, in which you have a adenine being there. So the adenine attacks, so internal 2 OH would be attacking the 5 of the OH being present, right? Or 5 phosphate being present of the exon. So remember about this, it is option number B which is the correct answer for this question. That is, a free 2 O of the internal adenosine would make a nucleophilic attack on the 5 P of the first base of intron. Remember about this, very very important. So this was about this question. Next question for you all. The question is that in the eukaryotes, the precursor of the microRNAs and small interfering RNAs are usually synthesized by which of the following? Quickly get the answer for this particular question, everyone. Take your time and get the answer for this question. Last question for the day. Quickly mark the answers. Done, everyone? Last question for the day. Quickly mark the answers. Okay, let's check. The answer for this particular question is option number D. That is, miRNAs and siRNAs are synthesized using RNA polymerase 2. You need to remember all these things. They are very very important, and time and again questions get repeated from this particular type of questions. Okay, I hope everything is clear to you. This was about for today's class. I hope all the things are clear to you. So just let me know what is your feedback regarding these type of concept mapping classes. In the similar manner, would you like to continue in the same thing, or should we switch to the practice sessions itself? Quickly let me know in the chat sections. We have 3 students in the class. Let me know whether you wish to have the practice sessions only, or these concept mappings would help you out. Yes, quickly everyone, just let me know, and accordingly we'll have tomorrow's class. Yes. Okay, we can switch to practice session itself, no worries. So from tomorrow we'll be having the practice sessions only. Okay, So this was about for this particular class. Thank you for joining in. Let's meet tomorrow. Bye everyone.